And today we have the great privilege of having our own Jeff Chamberlain speak to us today. He is the McCaw Endowed Chair in Muscular Dystrophy here in the Department of Neurology and Neurogenetics. Jeff went to undergraduate at Rice University in Houston and studied biochemistry there. He then went on to do his graduate work in his PhD in biochemistry here with Dr. Stephen Hauschka, and then a postdoc at Baylor before taking an assistant professorship at the University of Michigan. And he was there at the University of Michigan for quite some time before coming to Seattle in 2000 to take on a professorship here in this department and in the Department of Neurogenetics. Jeff's been a leader in muscular dystrophy field for many years. He has a 42-page CV, which is quite impressive, and is on many, many NIH study panels and national organizational leadership panels. So today I will let him take it away from here. Thanks.
Thank you. So it's a pleasure to be here. So I thought what I'd do today is focus mainly on research we've been doing in our group and in collaboration with many other people here at the University of Washington and Seattle on trying to develop therapies for the muscular dystrophies. Most of our work is focused on Duchenne muscular dystrophy, which is the most common of the muscular dystrophies. And a lot of this has recently been funded through our Wellstone Muscular Dystrophy Research Center. So I'll give you just a very brief introduction to the Wellstone Center for those of you that are not familiar with that. And then I'm going to talk about preclinical work we've been developing to try to move forward with gene therapy for Duchenne muscular dystrophy. And then I'm going to close out with some recent work we've been doing to try to develop potential next generation therapies using gene editing technologies. In the interest of full disclosure, I'm required to point out that I am on the scientific advisory boards of Solid, GT, Sarepta, and Akashi. So the Wellstone Muscular Dystrophy Research Center, which is centered here in Seattle, was funded by an NIH grant. It was set up through an act of Congress, mainly through a lot of lobbying, I think, by parent organizations and a lot of patient advocates in the muscular dystrophy field to try to increase NIH support of finding therapies for the muscular dystrophies. And we were fortunate to receive one of these centers here in Seattle. I'm the current director of the center, and Stephen Tapscott, who has a joint appointment here in neurology, is the co-director. And basically what this center is focused on is collaborative translational research. There are 4 entities that are part of the center: the University of Washington, the Fred Hutchinson Cancer Research Center, Seattle Children's Hospital, and also the University of Rochester. The goal, as stated here, is to accelerate preclinical research into the muscular dystrophies and to begin to move those findings into the clinic. So it's focused very much on translational research. There are 2 main disease targets that we work on. The first is on Duchenne muscular dystrophy, where the focus there is primarily on advancing gene therapy methods in preclinical animal models to try to move towards clinical trials. And there's also a lot of work on FSHD dystrophy, which involves development of new mouse models, adapting some of the gene therapy approaches that we've been applying to Duchenne, and also importantly to develop a clinical infrastructure for clinical trials related to the muscular dystrophies and to explore biomarkers and immune-related mechanisms in FSHD. So there are 2 main projects that form the center. Project 1 is a gene therapy project. Project 2 is more clinical studies. But I want to comment briefly on some of the core laboratories that are part of this center that are designed to support research, training, and communication. We have a viral vector core, which is focused primarily on AAV and lentiviral vectors. And the funding through the Wellstone Center is intended to support vector development for neuromuscular disease research, although we have mechanisms outside the center that we could potentially collaborate with other people that aren't necessarily interested in muscle diseases. There's an investigator development and training core, or IDPO core, which is focused on enhancing research and communication across the entire 5-state WWAMI region. to try to bring together both investigative students and patient communities. We've tried to integrate this fairly closely with the Department of Neurology, and particularly with the T32 training grant. And it also provides a growing and increasingly important interface between researchers and clinicians here in the Seattle area in particular, particularly with the muscular dystrophy clinics, both at Children's Hospital and at the University of Washington. And that's coordinated by Leo Wang, who is serving a really important role trying to bridge this basic research to clinical application gap. And he's been provided with some space by the department to help facilitate that. So we move on a little more towards the research side of things. So I'm going to focus today on Duchenne muscular dystrophy, which is the main focus of work in our lab. Most of you are probably pretty familiar with DMD. It's thought to be the most common lethal genetic disorder of children. It's inherited in an X-linked recessive pattern, and it's caused by mutations in what's thought to be the largest gene in nature, known as the dystrophin gene or the DMD gene. And biochemically, the disease is characterized by essentially a complete absence of the dystrophin protein in Duchenne muscular dystrophy. So it's a genetic null disorder. But there are milder versions of the disease, typically called Becker muscular dystrophy, in which case the mutations allow for production of small amounts or smaller size than normal dystrophin. So these are partially functional dystrophins made at low levels, and that leads to a milder clinical on-course. And this is important for the development of gene therapy because the observation that some patients who had very, very mild cases of the disorder actually had gene deletions that removed pretty large pieces of the gene. And that turned out to be important for the development of miniaturized versions of the dystrophin gene, which we really need to have for gene therapy. And I'll go into that in a little more detail, but keep in mind that the native gene is over 2 million base pairs in size. So the basic characteristics of the disease are progressive and ongoing muscle wasting, and most of the patients die from respiratory failure, although as some of these kids are starting to live a little longer these days, they're increasingly dying also of cardiac failure. There's a variety of good animal models for the disease. Most of the work that's been done over the years has utilized either the mouse or the canine model of the disease, although very recently there's been actually a rat model that was just published, a porcine model, and a non-human primate model. But the latter three models have not received a lot of work. And this is just sort of an organization of the gene. It has 79 exons in the muscle isoform of the gene. Again, it spans over 2 million base pairs of the human X chromosome, which takes up over 1.5% of the chromosome. And the gene is so large that cytologically it can be seen under the microscope. So what are some of the challenges for trying to develop a genetic therapy for Duchenne and Becker muscular dystrophies? The initial challenge that we faced for many years was just the basic concept of how can you possibly deliver a new gene to all of the muscles throughout the body in a safe and effective manner. And that's really come a long way through the development of vectors that have been derived from different viruses. And the idea there is that you remove the different viral genes that can lead to pathology and replace them with your gene of interest. So rather than a virus infecting a cell and causing disease, you're using the shell of the virus to deliver things into cells that then drop off their payload, in this case the therapeutic dystrophin gene. But can you adapt this system to deliver a gene so large, such as the dystrophin gene, which is over 2 million base pairs? Most of the vectors that have been developed in the gene therapy world typically are small vectors. They're derived from fairly small viruses, and they have a very limited carrying capacity. And that gets us back into this concept of trying to develop smaller and smaller versions of the dystrophin gene that could still be functional in preventing or reversing features of muscular dystrophy. So my lab has been working on this for well over 25 years, and we're finally at the point now where we think the methods are working well enough that they're starting to move into clinical trials. And in the future, and I'll come back to this hopefully if I have time at the end of my talk, we're starting to look at sort of next generation technologies which would involve, rather than trying to deliver a small synthetic version of the dystrophin gene for therapy, to actually use gene editing technologies to try to correct the mutations that are causing the disease. So the two main approaches that I'll be talking about are sort of the one that has been with us, I've been working on for a long time, which is body-wide dystrophin replacement, and this involves using a vector derived from adeno-associated virus, or AAV, to deliver miniaturized dystrophin genes that we named microdystrophins because they're so small. Our original, probably the major publication in this area was back in Nature Medicine in 2004, where we were able to show for the first time that a vector could be used to deliver genes throughout the body and target muscle at high efficiency. This was highly dependent on these smaller miniaturized dystrophin genes to be carried by the AAV vectors, the so-called microdystrophins. And as I'll show you a little bit of data on that, these microdystrophins, because they're so much smaller, they don't make a full-size protein. They make a protein that's only about 1/3 the size of the normal dystrophin protein, but they're highly functional, but they're not 100% functional. You do take a bit of a functional hit in using such a miniaturized version of the protein. An advantage of this approach, though, is that we feel that since we're replacing the normal or the endogenous dystrophin gene with a synthetic version, that this is something that should work in all patients regardless of their underlying genetic mutation. And that's fairly important because Duchenne muscular dystrophy is a new mutation syndrome where all patients essentially have a unique mutation. So there's not a common mutation that everyone has. The emerging research area that I'll talk about at the end is based on gene editing technologies that have been developed over the last 4 years using the CRISPR-Cas9 system. There was a series of papers published last year in Science by Eric Olson and others showing the potential use of this approach for Duchenne muscular dystrophy. Nick Bankston in my lab has a paper in Press that extends those technologies, and the advantages potentially of the gene editing approach as opposed to a gene replacement approach is that you have the potential to directly correct the genetic mutation, which would theoretically allow you to produce the full-length natural dystrophin protein. But that's highly dependent on the mutation that each patient has, and I'll come back to that later. So some patients have mutations that may not be correctable. Others have mutations that would be correctable only to make a partially functional protein and things like that. But in theory, a lot of these mutations can be either fixed or bypassed, and the technology has, in some ways, the potential to be a permanent treatment.
Excuse me.
Yeah.
Does the dystrophin molecule turn over very much, or is it?
It does turn over, like all proteins, but it has a fairly long half-life compared to most proteins. But it's still its half-life is estimated to be around 1 month. So any type of therapy you're going to need to do, you want to have production of this protein for many years. So you need a stable system that's going to continue to produce the protein. Yeah, that's all right. The other current limitation of the CRISPR-Cas9 system is the efficiency is currently quite low, and there are a number of safety concerns, but it's still an exciting and emerging technology. So both of these methods, whether it's gene replacement or gene editing, really rely on the use of adeno-associated viral vectors. And an advantage of AAV vectors, there's a number of advantages, there's some disadvantages, but AAV vectors, when they enter cells, they don't integrate into the host chromosome, so they're not significant worries about insertional mutagenesis, which had been seen in some early gene therapy trials with retroviral vectors that do insert. They exist as a stable episome within the nucleus of cells, but that has advantages and disadvantages. It works quite well in non-dividing cells, such as muscle or in neurons. It doesn't work so well in dividing cells. There are many different serotypes of AAV. There's at least 100 naturally occurring serotypes that are known, and there's been hundreds more that have been generated in the laboratory through DNA shuffling technologies. Many of these are highly tropic for muscle, so they're good for muscle gene therapy. There's no known pathology that's ever been associated with adeno-associated virus, so even the wild-type vector appears to be quite safe. Most people, if not everyone, have been infected with AAV at some point in their life. Again, our lab and more recently many other laboratories have shown that you can achieve systemic gene delivery throughout the body, and particularly to striated muscle, using vectors derived from AAV. Some of the limitations of the vector technology: it has a very small cloning capacity, so the wild type AAV genome is only 5,000 base pairs, and if you recall, we're dealing with a 2 million base pair gene. Fortunately, the dystrophin cDNA itself is only 14,000 base pairs. About 11,000 base pairs are protein coding, but we still need to cut that down significantly to fit it into these AAV vectors in order to deliver them to muscle. Some other disadvantages of the AAV vector system is they typically show very poor ability to get into stem cells, so it's not a great stem cell manipulating vector, and it's rapidly lost from dividing cells.
Any antibodies, Jeff, when you infect animals or man?
Yes, so that's another significant limitation right now. Certainly, when you deliver these vectors to any animal or to patients, you get very high levels of neutralizing antibodies that come up. So current technologies really limit you to a single treatment, and there's a lot of work going on to try to find ways to get around that. But perhaps more of a serious issue is that approximately a third of the patients that are out there are known to already have neutralizing antibodies against AAV due to a prior infection, and at least in the early stage clinical trials, those patients will probably be excluded from enrollment because of that. So that's a significant problem. So how do you make vectors based off AAV vectors? The technique, there's a lot of variations on this. A typical technique, and the one that we use in our laboratory, is uses a 2-plasmid or sometimes a 3-plasmid co-transfection system. The first plasmid has your therapeutic gene of interest. In our case, that would be the dystrophin gene, with the gene regulatory cassette up here to drive expression, typically in a tissue-specific manner. So in our case, we use muscle-specific promoters, and this has come out of a long-standing collaboration with Steve Hauschka's lab in biochemistry, who has developed really the best gene regulatory cassettes in the world for muscle expression. And then those are co-delivered with a plasmid that has various helper genes that you need for replication and packaging of the AAV vector. So these are, there's only 3 known genes in the AAV genome itself, but there's also a couple of adenoviral genes that you need for high level production of these vectors. So we transfect those into human 293 cells, and that takes your therapeutic gene of interest and packages it up in an infectious shell derived from the capsid protein of the AAV vector, and that can then be delivered to a variety of cells or tissues depending on your application. So here's an example of the use of these AAV vectors. This is a study we published about 10 years ago. This is looking at the expression of the dystrophin protein using antibodies that we generated against dystrophin. And this is what you would see in a wild-type mouse, just looking at a variety of different muscles, forelimb, masseter muscles, the heart, the diaphragm. And dystrophin is seen as a protein that binds to the interface of the muscle cell membrane. In the dystrophic mouse models, they're generally characterized by the absence of dystrophin. You do see occasionally these rare revertant dystrophin-positive myofibers. We usually try to find one to put in the image just to give you some contrast there, but essentially it's a dystrophin-negative animal. But once if we treat these mice through an intravenous infusion of these AAV vectors, we can restore a high level production of the dystrophin protein to all the striated muscles of the mouse. And over on this side here is a low power image of a gastrocnemius muscle from an animal that had been infused with a single dose of the AAV microdystrophin vector 1 year earlier. So as you can see, every myofiber in this muscle is staying positive for dystrophin even a year after the infusion. Yes?
You said that the coding region of the dystrophin gene is approximately 10 kb or so?
Yes.
But you could only get 5 kb in your vector.
Right, all right.
So this is a highly truncated protein?
Yes, it is, yeah. So I'll come back to that in a minute and give you a little more detail on that. So looking at, and we've published a number of papers looking at the functional outcome, we see a very significant impact on the dystrophic phenotype. It depends a little bit on the age at which you treat the animal, but in general we see a complete halt to the dystrophic process. There's no more ongoing necrosis or myofiber regeneration, and we see typically a normalization of the muscle histology and a significant increase in strength. However, I do want to point out a few things related to that. In this case, this is a functional study of the diaphragm muscle, which in the mouse model of Duchenne is the most severely affected muscle. And in this case, we can measure the specific force generating capacity of the diaphragm muscle. It's a sort of an indirect measurement of the strength of the muscle. And this black bar here just gives the readout we get from wild-type diaphragm muscles. The blue is the dystrophic model, and the red is a treated mouse. So as what you can see, we see a very significant increase in the strength of the diaphragm muscle, but it's not completely back to normal. And that's presumably a limitation of these miniaturized dystrophin proteins. On the other hand, another assay we look at is the susceptibility of different muscles to what's known as contraction-induced injuries. So it's thought that one of the major injurious events in particularly when Duchenne muscular dystrophy is exercise-induced injury. When muscles are undergoing lengthening contractions in particular, the force that's put on the muscle fibers can cause injury, which can lead to focal necrosis of the myofibers in the region of that injury, and then it's in skeletal muscle at least that's repaired through muscle regeneration processes. So we've set up assays in our laboratory where we can measure the force development of different muscle types in response to increasing lengthening contractions that are typically injurious. So the black bars here show the level of force development in a wild-type muscle after increasing stretches of the muscle. The blue line is what we see in the dystrophic mice, and the red line is what we see in mice that have been treated with a microdystrophin. So there's almost a complete protection from contraction-induced injury, and that's we feel a really critical thing of these microdystrophins. Even though they're not necessarily restoring full strength to the muscles, they are halting ongoing injury and damage from exercise-induced injury. There's another assay you can look at is just to measure the serum creatine kinase levels, sometimes known as CPK levels, that are very elevated in patients with Duchenne muscular dystrophy. And again, this is looking at a wild-type mouse, an MDX mouse, the mouse model for Duchenne muscular dystrophy, and then a treated dystrophic mouse treated with our microdystrophin. And what we see again is a 90% correction of the deficit between wild-type and untreated mice, but it's not 100% back to normal. And again, this is presumably a limitation of the microdystrophin design, and that's led to more recent work in our laboratory asking, are there ways to tweak the structure of these miniaturized dystrophins in order to maximize the functional capacity of those molecules? And so this gets back, maybe I should have put this up a little earlier. This is it's kind of a cartoon showing the domain structure of dystrophin. I'm not going to go into detail here, but dystrophin is essentially a long rod-like molecule that has a variety of different functional domains, actin binding domains, and then a variety of protein-protein interaction domains down at the carboxy terminus. And initially based on observations in patients that had large deletions in their gene, in other words, deletions that would remove significant parts of the coding region of the gene, and then looking at those deletions in patients that had very mild cases of the disease, it became clear early on that a significant part of the central part of the dystrophin molecule was not all that critical for its functional capacity. So we've extended that by making a variety of synthetic deletions in the laboratory in the dystrophin gene, removing different functional domains, and I'm only showing a subset of those. And those led early on to the observation that the carboxy terminus did not seem to be very important for the function of dystrophin, but large amounts of this central rod domain could be deleted. And so we've continued to sort of mix and match different subdomains to ask, what is the optimal functional capacity we can extract from these molecules while keeping them small enough to be carried by this AAV vector system? And some of the assays that we do to screen some of these newer microdystrophins are one, just an expression assay, and what we've noticed is that some of the mini dystrophins that we put together. They're expressed quite well initially after you inject the vector into mouse muscle, for example, but some of them are not that stable and they go away over time. So you see, like this one here is not expressed very well by this Western blot. These weren't expressed very well, but others are expressed quite well. We also do immunostaining to look at a large number of proteins that are known to interact with dystrophin, to ask, are we able to reassemble the normal multi-protein complex that the dystrophin assembles in muscle? And most of the early generation microdystrophins that we developed in our laboratory, and some of which have been developed by other laboratories since then, don't restore expression of the entire dystrophin glycoprotein complex, but our newest generation microdystrophins are able to reassemble all known proteins that associate with dystrophin. And then sort of the ultimate assay that we do is to ask the question of, if we deliver these microdystrophins to an animal model of muscular dystrophy, usually we use the mouse model, are we able to not only get stable expression and assemble the dystrophin complex, but how good are these proteins at restoring normal strength to the muscles? And again, one of our best microdystrophin we had come up with even just a few years ago is this one here that we sometimes call microdys H3. It was only able to, in this case, to develop about half of the normal or 2/3 of the normal specific force generating capacities that we see in wild type diaphragms. But one of our new microdystrophins in particular is almost able to restore normal strength of the muscle. So this is the version of microdystrophin that we're moving forward with and doing larger animal studies and trying to get into clinical trials. Here's just another example of different assays that we use. This is work done by Catherine Hollinger in our lab to look at the normal activity of dystrophic mice. One of the things that you see in the mouse models for muscular dystrophy is the mice are very easily tired. If you exercise them on a treadmill or you let them run around their cage, they'll often just stop moving and sit in the corner, so they tire very easily. And here's an example of just looking at the overall level of activity in an activity cage that uses a light beam to monitor their movement. Just treating these mice with saline gives a certain level of activity, but if you exercise them and then measure their activity, you can see it's significantly reduced, how much they're moving around the cage. But if we treat with a microdystrophin, we see not only the phenotypic increase in physiological increases I showed you earlier, but we see increased activity that's not nearly affected nearly as much by activity. Another thing we've seen is we've done some long-term time points and looking at a couple of different microdystrophins, in particular this one here, which is the current best one. We see very stable expression after delivery to muscle, even 2 years, which is the longest we've looked at to date. So the expression seems to be pretty stable. We and others are doing a number of studies now in the dog model of Duchenne muscular dystrophy. It's a larger animal model that hopefully will better predict outcomes that you might run into in the clinic. And here's an example of a study we did with Zhejiang Wang and Stephen Tapscott over at Fred Hutchinson Cancer Research Center. In this case, this was an intramuscular injection. Zhejiang did, I believe, between 60 and 100 individual injections to try to saturate a muscle. This was before we were getting heavily into the intravascular infusion methods. And looking 2 years after the infusion, we saw a very stable expression of these microdystrophins in the canine skeletal muscle. This is an untreated limb in the same dog where you get much greater histopathology and no dystrophin expression. And this level of expression was also shown to restore expression of other members of the dystrophin glycoprotein complex. So these are dystrophin-associated proteins here. So 2 years is the longest we looked with this type of assay. We saw stable expression and a significant increase or restoration of normal morphology in those dogs. More recently, we've been starting to advance towards systemic whole body delivery, and some of these studies have been done in collaboration with Dongsheng Duan, who's at the University of Missouri and has a very large colony of the dystrophic dogs. This is work that was funded by Solid GT that we're working with, using Microdystrophin, our best microdystrophin, driven from a muscle-specific promoter, CK8. This is the strongest one we know of that was developed, again, in Steve Hauschka's lab using the AAV9 serotype. This time, we typically here we use AAV6, but AAV9 has some advantages. Very high dose by IV infusion. And I don't know how well this shows up, but basically this is looking at 3 different time points out to 6 months after the infusion, and we have what appears to be stable and widespread dystrophin expression throughout the skeletal muscles of these animals. And more recently, Dongsheng has looked at one of his dogs sacrificed 15 months after the infusion, and the microdystrophin is still widespread throughout the body even at 15 months. So to summarize, hopefully what I've shown you is that some of the newest generation microdystrophins that we've been able to develop are able to assemble the entire dystrophin glycoprotein complex. They completely halt ongoing necrosis and regeneration in dystrophic muscle. They are highly protective against contraction-induced injury, and they show a significant, although not a complete, restoration of the normal strength to those muscles. Work, again, that I just showed you and has also been repeated in some other laboratories shows that delivery of the microdystrophin proteins using the AAV serotype 9 vectors is able to lead to systemic delivery even in these large animal models, which are golden retriever dogs. We haven't seen any microdystrophin expression through at least 1.5 to 2 years, depending on the delivery route we've used. Importantly, AAV vectors have been used in well over 100 human clinical trials without any significant adverse events. And there's a very exciting trial ongoing right now. This has not been published yet. It's ongoing at Ohio State University by Jerry Mendel. They've treated, I believe, around 15 patients with the most severe form of spinal muscular atrophy. These kids were treated with a very high dose of AAV vectors. Similar doses that we would like to use in the Duchenne trials. None of those kids have had any adverse events, and many of them are now alive significantly longer than they would have been without any type of treatment. So this is really validating the use of AAV vectors in neuromuscular disorders, showing that high-dose delivery can be safe and effective. And based on all of these things, there are now a number of clinical trials of the AAV microdystrophin that are either beginning or being planned by several groups. So I'll outline the groups that I know of that are moving in this direction. So Solid GT is a biotech startup company based in Cambridge, Massachusetts. They approached me about 2 years ago. This is a company that was founded by a family that has a child with Duchenne muscular dystrophy, and they were able to solicit donations from a number of their friends in the financial services industry, and they are moving very rapidly towards human clinical trials. They have licensed the AAV microdystrophin technology that was developed here at the University of Washington, and they're about to submit an IND application to the FDA to begin a human clinical trial, hopefully this year. There's a couple of other groups moving in a similar direction. Again, at Ohio State, at Nationwide Children's Hospital, Jerry Mendel has proposed doing a similar trial. Genathon, which is funded by the French Muscular Dystrophy Association, has proposed doing a clinical trial of microdystrophin. And most recently, Pfizer, particularly their gene therapy unit based in London, has jumped into this area and is proposing a trial also. So I can just tell you a little bit about some of the similarities and differences between these groups. So again, Solid GT is the group that we've licensed our technology to. They're hoping to use AAV9 with Hauschka's muscle-specific gene regulatory elements in our microdystrophin 5. They're proposing to do a systemic whole-body delivery in kids around the age of 7 to 15. It hasn't been set in stone yet. It's still negotiating with the FDA a little bit about how to do that trial. Jerry Mendel is proposing to do a similar trial with an AAV serotype that he identified at Ohio State in rhesus monkeys. Interestingly, he is planning on using one of our earlier generation microdystrophins that we published about 15 years ago. We feel that our new ones are significantly better than that, but he's going ahead with the older version. Based on his success with infants with spinal muscular atrophy, he's proposing to treat 3-month-old Duchenne boys with this technology. That has not been approved by the FDA. I don't know if they're going to go for that or not, but it's an interesting concept. Genethon in France is proposing to go ahead with a trial using AAV8. They also are planning on using our oldest generation microdystrophin that again, we feel has been replaced by significantly better ones. They're proposing a single limb perfusion technology rather than a whole body delivery. And then more recently, Pfizer has bought into the technology developed by Jude Samolsky and colleagues. They're also using a very old microdystrophin, very similar to our early generation clones. But beyond that, I haven't heard much detail in terms of age of patients or route of infusion. So we'll see how all of this goes. In terms of immune suppression, the current plans are to prophylactically treat all the patients with prednisone, which is a mild immune suppressive, and the Duchenne boys are typically on prednisone anyway. There are various groups exploring stronger immune suppression strategies that might block the production of antibodies, as we talked about earlier, which would be required in order to repeat the dose. Whether you would need to repeat the dose or not is a little unclear. As I think I've showed you, we can achieve whole body delivery with a single dose, but the AAV vectors, if you keep in mind, are episomal vectors. They don't integrate into the host chromosome. They don't get into muscle stem cells. And so our feeling is that these vectors would probably fade away over time through natural turnover of skeletal and possibly cardiac muscle tissue. So there's a possibility you'll need to repeat the administration. We don't know how often. It could be every 10 years, maybe longer, maybe shorter. But I suspect at some point you would want the ability to repeat the administration, and that would either require immune suppression at the time of delivery or perhaps switching to different serotypes of AAV that would not be neutralized by the initial protocol. So let me just close out a little bit by talking about sort of next generation technologies, and then that's moving on instead of using gene replacement to move towards gene editing to try to either correct or bypass the mutation in the dystrophin gene. And so CRISPR-Cas9 is a very interesting technology that has been shown to enable gene editing. And in theory for dystrophin, it should allow production of full or nearly full-length dystrophin, again, depending on the mutation and how you go about trying to correct that mutation. And I mentioned there were some papers published last year in Science showing the feasibility of this approach, and we have our own paper currently in press right now that has significantly extended those technologies. So the focus in our lab is to try to come up with ways to boost how much dystrophin is being made. A real limitation of the current CRISPR-Cas9 approach in muscle is that the efficiency has been very low. It's also problematic using these AAV vectors, and that most of the groups working on CRISPR-Cas9 approaches are using promoters that are active throughout the body, so you get ongoing continuous gene editing in every cell of the body. Working with Steve Hauschka, we've been able to modify this to do it in a muscle-specific manner so that at least we're only producing the gene editing nuclease, the Cas9 enzyme, in muscle tissue. And we've been trying to expand the mutational context to make this technology potentially applicable to all or most of the mutations that can lead to Duchenne muscular dystrophy. Just a very brief overview of the CRISPR-Cas9 system. This is a bacterial system that was identified, I don't know, 4 or 5 years ago. Initially by Jennifer Doudna down at Berkeley, and it has 2 parts. It uses a Cas9 enzyme, which is a nuclease that can cleave DNA, and it's guided to specific sites based on so-called guide RNAs that are able to anneal with the genomic DNA and bring in the Cas9 nuclease to induce typically a double-stranded break, although there are versions of Cas9 that will induce single-stranded breaks as well. And once you get these double-stranded breaks in your target region of your gene, those can either be repaired by non-homologous end joining, which is associated with small insertions or deletions, some of which might repair your mutation if you targeted the mutation right there for the Cas9, But there's another method you can use where you can bring in a donor DNA strand, and based on this double-stranded break in the DNA, you can bring in a repair template through homology-directed repair and get a complete correction of the gene rather than a bypassing the mutation. So we've been exploring this. This is work that's been led in my lab by a great postdoc, Nick Bangston, using two different versions of the Cas9 enzyme and delivering those with AAV. So now, again, we've run into some issues with the AAV technology in that it has a very small cloning capacity. And the Cas9 enzymes, together with the guide RNAs that you would need and the gene regulatory cassettes, can be very difficult to fit within this 5 kb cloning capacity of AAV. So in some cases, you have to split everything up into two different vectors. In other cases, with smaller versions of the Cas9 enzyme, you can just barely fit everything into a single vector. But one of the challenges, again, for Duchenne muscular dystrophy, this is from a paper we published a long time ago. I can barely remember it. But we went through a collaborative effort and screened over 700 patients for genomic deletions. And each one of these horizontal lines here represents the extent of the deletion that was found in a different patient with Duchenne or Becker muscular dystrophy. So this is, you probably can't see it very well, up at the top is the exon structure of the gene. So this just represents how many exons were missing from each patient. The bottom line here, there's two things. One is about 2/3 of all patients have a genomic deletion, so they're missing parts of the gene that you're not going to be able to repair. And secondly, these deletions are distributed all over the gene. So in terms of the way the CRISPR-Cas9 system works, to try to fix these mutations, you're going to have to direct your Cas9 enzyme to basically almost any part of the gene, depending on which patient you're looking at. So this would really be turning into a personalized medicine approach to try to develop a vector for each different mutation. Now, you can tend to drop these different deletions and mutations into different categories. So some people argue that you might be able to get away with 10 to 20 different vectors, but that's a lot of different vectors that we're going to have to go through FDA approval and preclinical testing in order to make this technology available for all the patients with this new mutation syndrome. So one way to get around that, the original papers that were published last year focused on a very simple point mutation in the original strain of MDX mice, and that proved to be relatively easy to correct. We decided to go after a different mutation further down in the gene in the so-called MDX4CV strain of this mouse model for Duchenne muscular dystrophy. And one of the differences is that, first of all, this mutation is in the region in this part of the gene, which is sort of referred to as the hotspot for mutations. 2/3 of the deletions in the gene are located in this part of the gene. Secondly, this is a mutation that can't be corrected by simply deleting the exon, as you could do with the original MDX mutation. So we took a variety of strategies. Again, this is led by Nick Bangston in my lab. One is to use CRISPR-Cas9 to clip out this entire genomic region, which would remove a small piece of the protein, but it's a part of the protein that's not very functional for its activity. And clipping out two different exons can restore an open reading frame to the messenger RNA and make a highly functional protein. Nick also looked at targeting the CRISPR-Cas9 system directly at the mutation in order to try to directly fix the mutation. Both strategies turned out to work. They're not equally efficient. This is just an example of following intramuscular injection into the MDX mouse. Hopefully you can see all these white circles here are dystrophin-positive myofibers in the skeletal muscle of two different treated mice with two different strategies. This was the complete deletion of 2 exons of the gene, about a 45 kb region of the gene. This was the approach to directly target the mutation or repair the gene. Both of them led to widespread dystrophin expression in the injected muscle. This is a control muscle here that is not showing dystrophin expression. By Western analysis, we could harvest these muscles after various periods of time and ask how much dystrophin are we actually making. Sometimes the immunofluorescent staining can be a bit deceptive. And we saw a lot of variability with this approach, partly because it's based on intramuscular injection, which is not the most reproducible method. But we saw levels of full-length dystrophin being made in these mice that range anywhere from one animal to another between about 1 and 23% of normal. So 1% is not that important, but once you get up into the range of about 20%, we feel that's enough dystrophin to have a very significant therapeutic impact on this disorder. So that's encouraging. Nick's now moved on to doing systemic delivery approaches, again with the muscle-specific promoter from the Hauschka lab, the smaller Cas9 enzyme together with these guide RNAs to try to direct the gene. These vectors have been infused intravascularly into these mice, and we've been able to look at striated muscles throughout the body, and what we can see is very significant levels of dystrophin throughout the striated musculature. I'm just showing you the heart here. This is a lower power image, a higher power image. As you can see, individual cardiomyocytes are making very significant levels of dystrophin, but hopefully you can probably also see there's a lot of regions that are not making very much dystrophin. So we're getting about a 35% efficiency here, as opposed to the simple gene replacement technology with the microdystrophin. We can close in closer to 100% efficiency. So this method is working, but the efficiency is a little low. And here's a side-by-side comparison using a similar dose of AAV vectors to deliver either the microdystrophin or the CRISPR-Cas9 system. And as you can see, we get much more uniform high-level expression with the simple gene replacement approach. CRISPR-Cas9 does work body-wide, but the efficiency is lower, and that's something that we're hoping to improve upon in coming years. So to summarize, the use of the muscle-specific CRISPR-Cas9 approach is that it limits Cas9 expression to muscle cells. We don't see any expression in livers, kidneys, spleen, non-muscle tissues. At least in this particular model of muscular dystrophy, we can induce expression of nearly full-length protein, which is highly functional. This leads to an amelioration of the phenotype, although interestingly, the functional correction we get is not as good as what we see with microdystrophin, presumably because we can deliver a lot more of the microdystrophin and it's more uniformly expressed because it's a simple gene delivery rather than a gene correction technology. Somewhat controversial, we were able to show that we could actually achieve homology-directed repair and get a complete correction of the gene, depending on the approach we're using, in these post-mitotic myofibers. So there's still room for improvement with this technique. We'd like to be able to get higher levels of dystrophin after gene editing. As I mentioned, the functional improvement we get is still kind of modest. It's significantly less than what we get with similar doses of the microdystrophin vectors. We're not able to target muscle stem cells, and this comes back to this issue of what happens 10 years down the road if you get normal muscle turnover and you have the myogenic stem cells regenerating muscle. They're not going to be bringing in more vector. So ideally, for a permanent correction, you'd like to extend these technologies to the myogenic stem cells. And I think there's still significant safety issues. This is an incredibly well-hyped technology. People are pushing to move it into the clinic, but there's still a lot of unknowns about off-target editing, cutting genes in the wrong cell type, and things of that nature. So I think there's a lot of safety issues that we need to carefully examine before we rush this technology into the clinic. So I'm going to stop there. I just want to thank the various many people that have contributed. I've only really been able to show a few of those. Nick Bankstrom has really led the Cas9 project. John Hall's contributed to a lot of this work, studying satellite cells, Cas9, FSHD work. Katrin Hollinger and Julie Credell are doing a lot of work on new generation microdystrophins, looking at the immunology. James Allen, Christine Helbert, and Luke Cody are doing most of our vector production and also run the vector core as part of the Wellstone Center. Matt, Maya, Darren, and many others have contributed to various aspects of this. And then some of our key colleagues here at the University of Washington are Guy Odom, Steve Hauschka, Joelle Chamberlain, Casey Childers, and Leo Wang. So happy to answer any questions. Thank you. Yep, Tom?
So what was the recent technology that was approved by the FDA?
Not sure what.
Were some families twisting the FDA's arm to get something approved?
So that was the exon skipping therapies.
Yeah.
So I didn't really get into alternative approaches, but one of the other approaches that is sort of gene therapy, sort of not gene therapy, is the idea of using small antisense oligonucleotides that can be delivered either orally or by injection into the vasculature. And the idea there is that these small oligonucleotides can anneal with splice sites in messenger RNAs and essentially mask those splice sites from the splicing machinery within cells and cause an exon to be skipped when the pre-messenger RNA is being spliced into the mature messenger RNA. So the idea there is it's a technology to try to trick the cell into jumping over a mutation and making a slightly shorter dystrophin protein that is missing the region around the mutation. So that's a technique that worked really well in mice, and it's been in clinical trials now for 3 or 4 years. There are 2 companies were competing to get a drug approved. One of which was BioMarin, and their drug was not approved by the FDA, and the second company was a Sarepta that was using a morpholino-based oligonucleotide, and their application to the FDA was initially rejected, but as you mentioned, after considerable lobbying and pressure from patient groups, the FDA decided to grant approval. So my opinion on the status of those drugs is that they clearly work to a certain extent. You can certainly see some dystrophin being made in patient muscles after you put them on these oligonucleotides, but the amount of dystrophin being made is very low. The main issue the FDA had with the Sarepta drug was that by Western blot analysis, they were able to only demonstrate that they were making about 1 to 2% of the normal levels of dystrophin, and many studies have suggested that's not enough to be effective. However, the kids that were in that trial, seemed to clearly be doing much better or significantly better than they would have otherwise. Only not a large number of patients, so it was controversial, but the drug is clearly very safe. It's limited to a single class of mutations, so it's only maybe about 10% of the patients, but it has been approved, and they're now ramping up production to try to apply that to more patients, to expand into new classes of antisense oligos to treat different types of mutations. And I know they're working very hard on the technology to try to improve the efficiency, because at the moment the efficiency is quite low, but it's the best thing that's out there so far. So we'll see what happens with that. Bruce?
So that's always fun to listen to, Jeff, because they're on the doorstep, perhaps, of providing an enormous amount of improvement in a disease which is lethal.
Mm-hmm.
That takes me to my question. I'm a little surprised, given the lethality of this disease and the incredible anxiety and pain that families suffer when they watch their children deteriorate, that we haven't been permitted to go ahead with some of these things.
Mm-hmm.
Can you comment on that? It strikes me that, I mean, if something bad happens, something bad is going to happen for sure anyway.
Mm-hmm. Right. It's been a tough situation. I think historically the FDA has been a little slow to jump on board with newer generation technologies. I mean, their major concern is safety, and so particularly a lot of the gene therapy technologies have come along very slowly in trying to interact with the FDA. However, my feeling now is that it's really changed a lot, that there have been so many gene therapy trials now that are largely have gone quite well, and the FDA is not raising nearly the hurdles and not requiring nearly as much preclinical testing as they used to in terms of gene therapy trials. Now, the situation with the exon skipping drugs is a little surprising, because there's no viral vectors there. It's a simple small molecule technology, and it's clearly been shown to be safe. There's a couple hundred patients have been dosed for several years with these oligonucleotide drugs without any adverse events. But the FDA was reluctant to approve those because they felt that a high bar had not been reached in proving efficacy. And they were really got hung up on this issue of doing very large, well-controlled studies with the large placebo groups, and many of the families in the Duchenne muscular dystrophy community are, they're highly educated, have been following these things for years. They're very opposed to placebo-controlled trials and things of this nature. And then the other thing that entered into the factor is that, I forget the name of the legislation, but Congress passed a bill about 2 years ago directing the FDA to really loosen up their standards in terms of rare disorders, lethal disorders like this. It's clear that message has not gotten to the regulators at the FDA. I mean, there was a major discussion about this at the FDA convened an open public hearing for the approval of the exon skipping drug from Sarepta, and there was a very contentious debate about whether they were following the mandate of Congress and allowing new drugs to be approved, and they kept going back to the 20-year-old definitions of we needed to see more placebo-controlled patients, we needed to see longer trials, and there was a U.S. senator that stood up and said, we passed a bill that make you stop doing this, and there was no response from the FDA. So we'll see what's going to happen. I think the most interesting thing about the approval that came out of that is that hopefully it sent a message to the FDA that they need to, for rare lethal disorders, they need to loosen things up a little bit. Now, I'm not proposing they completely eliminate safety standards or things like that.
No, of course you're not, Jeff.
Yeah,
I mean, I take your point. What would be the earliest that you might see a result from your trial?
It's difficult to say. If everything goes well with the approval process, we may be able to start treating patients later this summer. How long it would be before you see a functional improvement is a little unclear. It's from different types of trials in Duchenne muscular dystrophy that have been done so far. One of the things that's coming out of that is that actually the reversal of the disorder and actually measuring an increase in strength or mobility could be quite a slow process. In contrast, when we do studies on our mice, we see a measurable improvement within a month or two. In the patients, it could take longer than that, and part of the problem is that the rate of decline in muscle function is somewhat slow in this disorder. Even over a 1-year period, patients don't lose a huge amount of strength. I mean, it depends on what age they are and what stage of the disease.
But it's not the same for all patients, right?
It is the same for all patients. Yeah.
So it's not mutation dependent?
There's some variation, but it's a surprisingly tight curve, and this is one of the things the FDA got hung up on, was which mutation, all this kind of stuff. But people presented data on hundreds of patients comparing their rate of decline over the years, and there's certain mutations that seem a little slower, and the main thing that people often look at is loss of ambulation. At what age do they have to start using a wheelchair full time? And there's maybe a 2-year spread on patients that aren't receiving other treatments. So it's reasonably predictable. And then you run into other issues that if the patient has already lost ambulation, what are you really measuring in terms of their improvement? And the easiest thing to measure is the production of the dystrophin protein, but that requires a biopsy, and you want to limit having to take biopsies. And the FDA at this point has not agreed to the idea that actually making dystrophin is a biomarker for effectiveness of the therapy. They want to see a clinical outcome, basically an increase in strength or prolongation of walking time, things of that nature. Yeah. Yes.
Are there plans for newborn screening?
Are there plans for newborn screening? Sorry, I forgot I was supposed to repeat the questions. Yes, there are plans. There are various approaches to newborn screening for Duchenne muscular dystrophy. The simplest one is to take a Guthrie blood spot and measure serum creatine kinase levels. That's been shown to pick up about 70 or 80% of cases of Duchenne muscular dystrophy with a very low false positive rate. With the increases in genomic sequencing technology, people are starting to explore, panels of exome sequencing, things like that. I think those are a little further away. But there's been some reluctance to implement newborn screening for Duchenne muscular dystrophy because of the cost and the fact that there's not a proven therapy available at this time. But I think a lot of people feel that if any of these technologies, either the exon skipping or the gene therapies that we're trying to develop, if those really start showing promise, there's going to be a major push to implement newborn screening for Duchenne and throughout the United States.
Or even like a 2, 4, 6 month check where your elevated CPK is going to have less false positives and higher.
But potentially, yeah. Most of the work I've seen has used the Guthrie blood spots because those have been collected anyway and all that. Yeah. Yes?
How does the immunomodulation, and immunosuppressive drugs affect the efficiency of delivery?
Well, I mean, I haven't seen any studies suggesting that there's an adverse impact of immune suppression. So, it doesn't change the efficiency of gene delivery or gene expression. The advantage early on, a lot of labs were running into problems with the vector itself eliciting a cellular immune response, and that was associated with a loss of gene expression over time. With newer methods of generating and purifying these AAV vectors, that response has largely gone away, even without immune suppression. So now the issue that most people are concerned with is this idea of eliciting neutralizing antibodies against the vector or having pre-existing neutralizing antibodies against the vector. And what happens there, if you're trying to do an intravascular delivery of this vector system and the patient has high levels of neutralizing antibodies against the vector or against the wild-type virus, that would neutralize the vector and prevent it from getting into the muscle. And potentially could be associated with an adverse event from this massive amount of antibody vector aggregate that's going to come out of that. So basically what's been seen is that the situations where gene transfer has not worked very well has generally been associated with an animal model or an individual that had high levels of pre-existing antibodies. So in the case of pre-existing antibodies, that's going to be a little difficult to deal with. Some people have proposed using plasmapheresis to try to greatly lower the amount of pre-existing antibodies. But the other approach is the idea of using an immune suppressive strategy that would prevent B-cell recognition of the vector in order to elicit production of neutralizing antibodies so that you could potentially repeat the administration down the road. I think ultimately the more challenging question is going to be patients that have already been exposed to AAV and already have immunity to the vector.
On that note, then, if you had, does the administration of the vector to naive, so people who don't have immunity to the adenovirus, how long is that's enough to generate a neutralizing antibody response to prevent future administrations?
Well, make sure I understood your question right. I mean, so there's two situations. One, you have patients that had a prior exposure to adeno-associated virus. Those patients almost always maintain fairly high levels of neutralizing antibodies. They may also have cellular immunity against the AAV vector. So those patients are going to be very difficult to treat. But even in a naive patient that has never been infected with AAV, once you deliver this vector to those patients, you are essentially vaccinating them against the vector. And so you're going to elicit high levels of neutralizing antibodies that would then neutralize a second dose and prevent it from being delivered throughout the body. So does that answer your question? Yeah. Yeah.
First, thank you for your great job. Do you define any stage of the disease that you think administering these drugs will help? Is there any stage like biopsy or by functional?
Yeah, so I mean the question is it, well, I guess there's two parts of that. One, is there an ideal age to treat, and is there a stage of the disease where it's too late? It's difficult to be sure about that. I can give you my opinion on it. I think the earlier you treat, the better. Because, Duchenne comes on somewhat slowly. If there's not a prior family history, many of these kids are not picked up until they're 5 or 6 years of age. So presumably, if we can treat these kids in the first few years of life, you would actually be preventing the disease from coming on. Now, once the disease has really taken hold, in particular once the kids have lost the ability to walk, that's associated with a significant loss of muscle mass. It's not so much that the muscle isn't working so well anymore, but you're losing muscle mass. It's being replaced by fibrotic and connective tissue and fat cells, things like that. So the gene therapy approaches are not going to eliminate fibrotic tissue. They're not going to get rid of adipocytes that have moved in. They're not going to grow new muscle. So what we hope to do is preserve whatever muscle mass and function is in the patient. So is there a stage where the technology wouldn't work? I'm not sure. I mean, there's stages when, presumably at any age of the disease, you should be able to halt any further deterioration. Whether you're going to bring back a lot of strength is we don't know yet. Different considerations perhaps for the heart, because the heart doesn't go through the necrosis and regeneration like you have in skeletal muscle. It's fairly well preserved, although the patients develop a very severe cardiomyopathy later in life. Whether that's, you could argue maybe in a patient that's 30 years old, you could rescue their heart function but not do a lot to the skeletal muscle, but it's all speculation at this point. Yeah. Bruce? Yeah.
Jeff, and I think we're probably towards the end of interrogating you. You made the point, and it's always been a nice biological curiosity what a huge protein this is.
Mm-hmm.
A gigantic gene and a giant protein that's created by that gene. And then you made the comment that a lot of Duchenne's is a new mutation.
Mm-hmm.
So is that just because of the size of this gene? And Tom may have an opinion about this as well. I mean, do huge genes just simply give you a lot more territory for a random mutation to muck up the functional properties of that protein?
Probably. I think that's what most people think. So the question is this is a new mutation syndrome? It's a very large gene. Do those two go hand in hand? And I think the answer is probably yes. It's the largest known gene, and there's just a lot of different things that can go wrong with that gene. And it's interesting, I mean, if you look at the deletions in the gene, as I mentioned earlier on, 2/3 of the patients have a genomic deletion, and those deletions can be anywhere in the gene. So presumably, if you assume, which probably isn't true, but if you assume there was a random rate of just deletions anywhere in the human genome, the bigger the gene, the more common you're going to find mutations. And then being an X-linked gene, you have that added impact that in males, if they get one mutation in the gene, they have the disease, whereas with autosomal recessive diseases, one mutation in a gene is usually not going to be associated with the clear disease case. So,
Jeff, you talked about the future, the golden age of neurology and neuromuscular neurology. Thank you very much.
Thank you.